Welcome to the Protein Room. I will be mainly talking about the hemoglobin and also talk a little about sickle cell anemia. Before we get started, let's gain a basic understanding of protein function. Proteins are the most abundant macromolecules found in cells, and they play many different roles. For example, the respiratory pigments hemoglobin and myoglobin bind to oxygen. Hemoglobin transports the oxygen, which is essential for cellular metabolism. Collagen and keratin are structural proteins associated with the skin, connective tissue, and hair of organisms. Actin and myosin are contractile proteins found in abundance in muscle tissue. Other examples are the immunoglobulins, which function in the immune system of vertebrates, transport proteins involved in movement of molecules across membranes, some of the hormones and the receptors, which regulate various types of chemical activity, and histones, which bind to DNA in eukaryotic organisms. What is hemoglobin? Hemoglobin is found in red blood cells. Hemoglobin are globular proteins that carry oxygen and molecules and carbon dioxide molecules throughout the body. Each hemoglobin protein structure consists of 4 polypeptide subunits, which are held together by ionic bonds, hydrogen bonds, hydrophobic interactions, and van der Waals forces. 4 polypeptide chains, each wrapped in a specific way around its own hemigroup, make up the hemoglobin molecule. These hemigroups contain positively charged iron, Fe2+ molecules, which can bind to oxygen molecules and transport them to various areas of the body. However, iron in the Fe3+ state cannot bind to oxygen. As the hemigroups bind or release their oxygen loads, the overall hemoglobin undergoes changes which alter their affinity for oxygen. Next, let's talk about the German chemist Emil Fischer. Around 1900, he determined the manner in which the amino acids are bonded together. He showed that the amino group of one amino acid can react with a carboxyl group of another amino acid during a dehydration reaction, releasing a molecule of H2O. The resulting covalent bond is known as a peptide bond. 2 amino acids linked together constitute a dipeptide, and 3 a tripeptide. Once 10 or more amino acids are linked by peptide bonds, the chain may be referred to as a polypeptide. Generally, no matter how long a polypeptide is, it will contain an amino group at one end and a carboxyl group at the other end. There are 4 levels of protein structure. First is the primary structure. The sequence of amino acids in the linear backbone of the polypeptide makes up its primary structure. This sequence is specified by the sequence of deoxyribonucleotides in DNA through an mRNA intermediate. The primary structure of a polypeptide helps determine the specific characteristics of the higher orders of organization as a protein is formed. Here, you can see an example of the primary structure. Next is the secondary structure. The secondary structure refers to a regular or repeating configuration in space assumed by amino acids aligned closely to one another in the polypeptide chain. In 1951, Linus Pauling and Robert Corey predicted an alpha helix as one type of secondary structure. Since then, the alpha helix model has been confirmed by X-ray crystallographic studies. It is rod-like and has the greatest possible theoretical stability. The helix is composed of a spiral chain of amino acids stabilized by hydrogen bonds. The side chains, also known as the R groups of amino acids, extend outward from the helix, and each amino acid residue occupies a vertical distance of 1.5 angstroms in the helix. There are 3.6 residues per turn. All proteins displaying an alpha helix are right-handed. Also, Pauling and Corey proposed a second structure, the beta-pleated sheet configuration. A single polypeptide chain folds back on itself, or several chains run in either parallel or anti-parallel fashion next to one another. Each structure is stabilized by hydrogen bonds formed between atoms present on adjacent chains. A single zigzagging plane is formed in a space with adjacent amino acids 3.5 angstroms apart. Most proteins demonstrate a mixture of alpha and beta-pleated sheet structures. Here you can see the beta-pleated sheets and the alpha helix. Here's a picture of Robert Corey and Linus Pauling. Next is the tertiary structure. This structure defines the 3-dimensional conformation of the entire chain in space. Each protein twists, turns, and loops around itself in a specific way. 3 aspects of this level of structure are most important in determining the conformation and in stabilizing the molecule. 1. Covalent disulfide bonds form between closely aligned cysteine residues to form the unique amino acid cysteine. 2. Nearly all of the polar hydrophilic R groups are located on the surface where they can react with water. 3. The nonpolar hydrophobic R groups are usually located on the inside of the molecule where they interact with one another and avoid interaction with water. It is important to know that the three-dimensional conformation achieved by any protein is a product of the primary structure of the polypeptide. The three stabilizing factors depend on the location of each amino acid relative to all others in the chain. As folding occurs, the most thermodynamically stable conformation possible results. Here you can see the ionic bonds and the polar forces and how they each have their own twisting and turning. Next is the quaternary structure. This applies only to proteins composed of more than one polypeptide chain, indicating the conformation of the various chains in relation to one another. This type of protein is called oligomeric, and each chain is called a subunit. The individual subunits have conformations that fit together with other subunits in a specific complementary way. Hemoglobin is an oligomeric protein consisting of 4 polypeptide chains. Most enzymes, including DNA and RNA polymerase, display quaternary structure. Here you can see the alpha chain, the beta chain, the heme, and the iron. And this is also what heme looks like. Now, let's talk about sickle cell anemia. Sickle cell anemia is a genetic disease in humans that is caused by an autosomal recessive gene and is fatal in the homozygous condition if untreated. It is caused by an alteration in the amino acid sequence of the beta chain of globin. Comparing normal and sickle cell globin and hemoglobin. So the template 3' to 5' Watson DNA strand. Normal beta globin has CTX, and sickle beta globin has CAX. And you can also see that in the blue, the changes are made in the DNA strand and mRNA transcript. Now, there's also a change in the first 9 amino acids on the polypeptide chain. In the blue, you can see the 3-letter amino acid codes are different, and the 1 amino acid codes are also different, as well as the amino acid classification. The changes are in the blue. Genotypes and phenotypes. If you don't have sickle cell anemia, your genotype is HbA-HbA or HbA-HbS, and your phenotype is normal, healthy, pain-free. If you have sickle cell anemia, your genotype is HbS-HbS, and your phenotype is low oxygen supply to tissue. You have pain, organ damage, strokes, and infections. Also, African Americans are more susceptible to sickle cell anemia. 1 in 500 African Americans have it. And now let's talk about carriers. If you have a father that is a carrier and a mother that is a carrier, one child will not be affected, two children will be carriers, and one child will be affected. Symptoms of sickle cell disease prevents oxygen from reaching the spleen, liver, kidneys, lungs, heart, or other organs, causing a lot of damage without oxygen. The cells that make up these organs die. Also, the red blood cells don't live as long as healthy red blood cells. Healthy red blood cells can live for 120 days, and sickle-shaped red blood cells live for only 10 to 20 days. When sickle-shaped red blood cells get stuck in blood vessels, patients can have episodes of pain called crises. Here you can see an example of the sickle cells getting stuck and how normal sickle cells are able to make it through the pathway. Also, organs damage and other complications often shorten people's lives by about 30 years if they have sickle cell anemia. I hope you learned a little bit about hemoglobin and sickle cell anemia. Thanks.